My name is Joshua Alper. I'm a biophysics leader in drug discovery for many years. Been working at small biotechs and large pharma companies. One of the real key drivers of drug discovery these days is DNA-encoded libraries. And DNA-encoded libraries enable one to screen billions of compounds in one tube against a single target. So it's really a massively high throughput screening technology. Traditional way of evaluating DEL hits is to take the compounds, then resynthesize those off DNA and quantify the binding of them. This process has some challenges associated with it. One of those challenges is that the library itself is made on DNA, and the DNA helps stabilize the synthesis procedure. If you're going to synthesize one of these on DNA compounds, it's done in aqueous solution. It's a whole different synthesis procedure than you would do if you're going to synthesize it off DNA. It becomes a challenge for the chemists after having identified molecules of interest that they have to come up with a new synthesis procedure often. And this really extends, or this can be a long lead time item. If you have the starting materials that you use to build your library, you can and you know the synthesis procedure that you used to build that library, you can replicate building that library with just resynthesizing the one molecule that the DNA-encoded library screen said that you would want to follow up on. And so you can accelerate the pace at which you make the compounds to validate by doing an on-DNA binding conformation. I think one of the great advantages of the on-DNA binding workflow is that you have this natural tag on it that you can then use to capture the small molecule. And so you can then use something like SPR, where you're not limited by capturing the protein and getting a very small signal amplitude. By capturing the small molecule and measuring the binding of the larger protein, you can get a much larger signal amplitude and therefore quantify the binding with really robust KDs. I think one of the really exciting things about on-DNA hit validation, particularly if it's being done at a large pharma company where you're cranking through lots of different possible targets, sometimes those targets are in complicated systems. Maybe they're on cells, or maybe they're out of cell lysates. Many traditional SPR techniques have difficulty making measurements, quantitative measurements on cells or in cell lysates. And I think that that's where a technology like focal molography really offers some additional promise in a DNA encoded library on DNA binding conformation workflow. There's a couple different reasons. So you want to always do your hit assessment in the most biologically relevant sample that you can possibly do. And so perhaps that's a cell lysate, perhaps that's in blood serum, perhaps that's depending on the target, just with a more biologically relevant buffer. I think that that's an important part. Also, a lot of effort can be spent into making purified protein to put into an SPR. And if you can avoid some of those steps where can lead to protein aggregation and keep that in a more biological system, a more biologically relevant system, then I think the likelihood of success out of a biophysical binding measurement is higher. As the low-hanging fruit in drug discovery gets used up, right, the targets that are less well understood, but that there's and what I mean by less well understood is that targets for which the mechanistic biology is less well understood, but that there may be strong genetic evidence for, become interesting new potential targets for drug discovery companies. And because there's not a lot of work that's been done on these, but that there's interesting genetic evidence, the likelihood that there are tools for those compounds, that there are binders, that there are known inhibitors, that there are known activators, is low. And so, as drug discovery companies dip further into the genome to find the new great target, I think that something like DNA-encoded libraries and on-DNA binding conformation, really provides an avenue to accelerate that kind of discovery.